Welcome to the Explainer. Okay, let's just jump right in because today we're looking at one of the most fascinating scientific puzzles of our modern age. We're totally surrounded by electromagnetic fields, or EMFs. They are literally everywhere. But for a long time, there's been this core scientific paradox right at the heart of our understanding of them. How could everyday low-energy EMFs possibly affect human biology? Well, the research we're unpacking today, which is driven by Dr. Martin L. Pall, finally gives us a really compelling, biologically plausible answer to this mystery. So, think about it for a second. The crucial point is this. The electromagnetic fields we run into daily, like low-frequency fields and microwaves, they're made up of low-energy photons. And these photons simply do not carry enough energy to heat up our cells or individually change the chemistry inside them. So without that thermal heat, how in the world do non-thermal biological changes actually happen? How did these invisible waves actually alter us? To really get this, we kind of need to think of the human cell as a fortress. The plasma membrane is this highly secure wall, and it's designed to keep the inside strictly regulated and protected from the outside environment. But the science shows us that something is getting past those walls. These low-energy, invisible fields are effectively breaking in, and they're doing it without knocking down the walls or burning the fortress down. Basically, they're using a key. And those keys are unlocking what we can think of as secret doors. These doors are called VGCCs, or voltage-gated calcium channels. They are literally electrical security gates embedded right there in the cell membrane. Now, under normal circumstances, they open and close based on the electrical voltage gradient across the membrane. But the research reveals that these specific channels are incredibly sensitive to electromagnetic fields. When they're exposed to them, these gates kind of glitch, and they just swing wide open. Now, here's the thing. This isn't necessarily a brand new, isolated discovery. Like Dr. Martin L. Pall points out, the answer to this question has been hiding in plain sight in the scientific literature. You see, because science is often so highly specialized, the clues were just scattered across different fields and decades of research. It was basically waiting for someone to connect the dots and realize the sheer volume of data pointing directly at these calcium channels. Let's look at the sheer weight of that evidence for a second. Dr. Pall reviewed 23 separate independent studies, and guess what? All 23 of these studies found the exact same thing. Electromagnetic field exposures act by activating these voltage-gated calcium channels. It doesn't even matter if it's extremely low-frequency fields, microwaves, or static magnetic fields. The primary target that reacts to the exposure is consistently the VGCC. And looking at this data really brilliantly illustrates exactly what happens when those doors open. Check out this data point from the literature, where they were testing bovine chromaffin cells when hit with a single nanosecond electric pulse. An incredibly brief exposure. There is a very rapid, almost instantaneous increase in intracellular calcium. It is so fast that it actually rules out a lot of other indirect, slower regulatory biological effects. The EMF hits, the VGCC gate opens, and calcium just immediately floods the cell. And we can see how this builds, right? Because this isn't just happening in some obscure, specific type of cell. The data here shows pulsed magnetic fields affecting human lymphocytes. Those are our immune cells. The influx of calcium through these channels alters cell proliferation and cytokine production. It's the exact same mechanism at play, altering the foundational behavior of our immune system's frontline defenders. Okay, let's break down this biological chain reaction step by step so we have a perfectly clear picture of the whole shebang. Step 1, the body gets exposed to an EMF. Step 2, the voltage-gated calcium channels in the cell membrane are electrically stimulated and they activate. Step 3, this opens the gates and a massive amount of intracellular calcium floods into the cell. And finally, step 4, this sudden influx of calcium binds with a protein called calmodulin, and that triggers a massive, rapid spike in the synthesis of nitric oxide within the cell. Now here is where the explainer gets truly fascinating. That spike in nitric oxide, it's essentially a biological crossroads. I actually like to call it the Dr. Jekyll and Mr. Hyde of cellular pathways. From this one single trigger, calcium flooding in and boosting nitric oxide, the cell can go down two drastically different paths. One path is physiologic. It leads to healing and cellular growth. The other path is pathophysiologic, and that one leads to severe damage and disease. Let's explore both, and we'll start with the good news. Section 1, the healing pathway, our cellular Dr. Jekyll. So how can EMFs actually be used for healing? Well, the most prominent medical example is using them to stimulate bone growth and repair fractures. And here's the specific biochemical sequence for that healing process. When nitric oxide levels increase in a healthy, physiological context, it stimulates the synthesis of a molecule called cGMP. That, in turn, activates something called protein kinase G. When this specific pathway lights up in bone tissue, it tells the body to increase osteoblast differentiation and maturation. In plain English, it literally signals your bone cells to multiply, mature, and rapidly repair damage. And the literature backs this up beautifully. Look at this entry highlighting human mesenchymal stromal cells. By applying a biphasic electric current, medical professionals are actively using this exact pathway to stimulate osteoblast differentiation and cytokine production. By deliberately opening those calcium channels with very specific electrical currents, we can trigger the jackal pathway to heal complex bone fractures. It's really incredible. Section 2, the damage pathway, or Mr. Hyde. But we do have to pivot our narrative here, because while that initial trigger can heal, in most contexts, it actually causes profound harm. The danger happens when that surge of nitric oxide meets another molecule inside the cell called superoxide. When these two mix, they form a highly destructive, potent non-radical oxidant called peroxynitrite. This is the toxic cocktail that sends the cell tumbling down the damage pathway. And this leads directly to a state of severe oxidative stress. You see, peroxynitrite breaks down into dangerous free radicals, including hydroxyl radicals. And these free radicals act like microscopic wrecking balls, basically bouncing around inside the cell, producing oxidative damage, tearing at cellular structures, and just wreaking absolute havoc on the healthy function of our biology. To understand just how serious this wrecking ball effect is, we can look at the evidence of DNA damage. Multiple studies using things called alkaline comet assays have shown that these EMF exposures can lead to single-strand breaks in cellular DNA. Now, just neutrally reporting the science here, the literature shows this damage is heavily linked to that peroxynitrite pathway we just talked about. We know this for sure because when scientists introduce antioxidants, which fight oxidative stress, or if they use inhibitors to block the creation of nitric oxide altogether, the DNA single-strand breaks are greatly lowered, or sometimes entirely prevented. Section 3, protecting our cellular health. So knowing this damage pathway exists, how do we actually protect our cellular health? Well, the paper proposes a few specific interventions. First, you can use calcium channel blockers. Basically, if you lock the VGCC doors, the calcium never floods in. Second, you can use NRF2 activators. These induce enzymes that degrade superoxide, which prevents that toxic peroxynitrite mixture from ever forming in the first place. And finally, using potent antioxidants, like phenolics, can help neutralize the free radicals if they do form, essentially stopping the oxidative stress right in its tracks. All of this microscopic biochemistry brings us to a really massive, real-world question. What happens to our cells in the digital era? We aren't just occasionally exposed to EMFs in a lab. We are bathed in them constantly, every single day. Specifically, there is a growing scientific concern around something called dirty electricity. And here is why that matters. Our modern digital devices don't just emit smooth, continuous waves. Digital technology is inherently based on rapid, square-wave transients. These are sharp, jagged bursts of electromagnetic energy. In the scientific literature, we already know that incredibly brief, high-intensity nanosecond pulses successfully blast open those VGCC secret doors. Dirty electricity essentially operates as rapid, continuous bursts of these exact transients. Which leaves us with this final, truly provocative thought to take away from this explainer today. As we completely surround ourselves with digital devices, smart tech, and wireless infrastructure, all of which emit these rapid transient fields, are we unknowingly, constantly unlocking the secret doors of our own cells? And if we are, which pathway, Jekyll or Hyde, are we triggering day in and day out? It's a question the scientific community is racing to answer, and it absolutely changes the way we look at every single device we own.